Get that. Okay. This is where we left off. And if you have two HbA and HbS heterozygotes, now we're talking about sickle cell anemia. So HbA is the normal hemoglobin molecule. Okay, HbS is the abnormal one. It's not necessarily a bad thing. So heterozygotes, in this case, when they're in an area that's infested with malaria and Anopheles mosquitoes, heterozygotes have the highest fitness because heterozygotes are protected to a certain extent from the parasite entering the red blood cells. So if you had two HbA here. Okay. Okay, so this is the normal hemoglobin. However, individuals with this phenotype, genotype and phenotype, are susceptible to the parasite. So in areas where malaria is endemic, these individuals are not going to be protected, and they're going to suffer very high rates of either getting very sick or dying from malaria. Okay? Now, okay. So HbS, these are homozygote recessive, and individuals who inherit two copies, one from each parent, end up with sickle cell anemia, and they generally don't live past their teens, and so their fitness is very low. Okay? Whether they live in an environment with malaria or an environment without malaria, having this genetic configuration is going to kill them before they can actually breed. Okay. This is the heterozygote that we talked about. So if two HbA-HbS heterozygotes marry, so let's just go back to these heterozygotes first. These heterozygotes in an area where malaria is endemic have the highest fitness. This is called heterozygote superiority. Okay. Heterozygote superiority means that the heterozygotes have the highest fitness. Homozygotes, normal, don't have very good fitness because they're susceptible to malaria. HbA, HbS homozygotes, because those individuals have sickle cell anemia. So they have low fitness too. Now, if you were to take these genotypes and transfer them to an area where there's no malaria, the highest fitness would be HbA, the homozygote normal. Because the HbA suffers a little bit in terms of oxygen transport compared to the normal, but it gives you advantage in being not susceptible to malaria. But if there's no malaria around, the top fitness goes to the homozygote normal. Okay? So, I just set the stage for that. So here's what's called a Punnett square. Okay. And we'll say at the top is the man. Oops. Hold on. And along the bottom, or on the sides rather, is the woman. Okay. Now, each of these individuals is a heterozygote. So for each square, you give them one chromosome. So for the man along the top, there will be let's see. Okay. And then okay. Okay. So, each of these are individual chromosomes. Okay? The man is going to either donate an HbA or he's going to either donate an HbS. The same for the woman. So each of these squares is then going to be turned into a diploid individual. Okay? So if we have two HbA matings, in other words, gametes that are HbA from the man and the woman, okay. Now, if we go across, we now see here's the woman's HbA. Right there. And in this case, the man's HbS. And so in this square, we end up with HbS. A, HBS, and that's the heterozygote. If we come down to the bottom rung, right here, so this is the woman's HBS, and if we pair her with a man's HBA, we end up with okay. So if you look at HBA, HBS is up here, right? And HBS, HBA is down here, but they're exactly the same. Okay, they're both heterozygotes. And then finally, we're going to get, in this corner, we have an HBS from the woman and an HBS from the man. And so we're going to get HBS, HBS, and this individual will have sickle cell anemia. Now, since each square is worth 1/4, right, it asks, how, given that HBS is recessive, what's the probability that the child will have sickle cell anemia? To have sickle cell anemia, you have to be homozygotic HBS. That means you have to be this. So what's the probability that you'll get an HBS child?
25%.
1/4?
25%.
25%, right. Okay, so. Okay. What's the probability that you'll end up with a carrier who actually carries the HBS allele but doesn't suffer the consequences of being recessive?
50%.
So 50%, that means we have this is 1/4 and this is 1/4. And so HBA, HBS is, okay. Okay, so that's 50%. And then, of course, there's a 1/4 chance for having the wild type with normal hemoglobin, HBA, HBA. Okay? Okay, so this is the same thing again. I've just made it a little easier. Right? Okay. Now, these are genetic recessive diseases. That means you need 2 copies of the mutated allele for the individual to suffer the consequences. In something like sickle cell anemia or, let's see, Tay-Sachs disease, a whole bunch of other kinds of diseases like this. And a good one was retinitis pigmentosa. Remember when I talked to you about the British colonists? There were like 15 put on this island in the South Atlantic. One of those individuals was a carrier for retinitis pigmentosa, 1 out of 15. Unfortunately, he managed to breed and pass on that gene, and eventually that allele rose in frequency. Just because of genetic drift, so that the population as a whole eventually developed much higher levels of retinitis pigmentosa because it was an inbred population. In other words, two individuals were probably more closely related to their ancestors because of that colonization event, and both the offspring might get unlucky enough to inherit 2 copies. Okay. Now, there are some human genetic diseases that are dominant, not recessive. In other words, all you need to do is inherit 1 copy, and the individual will suffer the effects. So a good example of this is called Huntington's chorea. And what it is it's there's been this replication of repeats within the middle of the gene that produces an abnormal enzyme that would normally break down some other protein. Because that protein isn't broken down and dispensed with, it builds up in the cells, and it's a neurological disease, so it causes brain problems, namely motor problems as well as psychological problems, all kinds of terrible things. It's adult onset, so it usually doesn't show up until an individual is about 35 years old. And then when they do, they're going to be a goner. And it's not a pleasant death. So, anybody know who Woody Guthrie is? This land is your land, this land is my land. Okay, he wrote that song. He's got a son named Arlo Guthrie. You might have heard of him. Well, Woody Guthrie inherited Huntington's chorea. Didn't know he had it. Then he started acting a little strangely, like some of his previous ancestors. I think it was on his mother's side. And then eventually he was diagnosed with Huntington's chorea. He was a little unusual in that he actually survived with the disease for about 14 years from the time he was diagnosed, which is unusual. Unfortunately, he had a bunch of kids. Kids from a number of different wives. And so none of the kids knew whether they had actually inherited the allele or not. Because it's dominant, there's a 50% chance that an individual who has one parent with the dominant allele will actually pass it on to their children. Because their children only need 1 copy and not 2. Okay? Fortunately for Arlo, he didn't inherit it. I'm not sure about his other kids. But anyway, it was passed on like this. So, cystic fibrosis is another recessive allele, recessive genetic disease. The CFTR protein normally transports chloride ions across cells of the lung. If the protein is not functioning properly, a thick mucus layer builds up in the lungs, and it also builds up in the stomach and interrupting kind of digestion. And So cystic fibrosis is another recessive disease. You need 2 copies. It's thought that individuals who are heterozygotic are protected from another disease called typhoid fever. Okay? So the heterozygote is protective. The homozygote means that, for the most part, individuals who inherit 2 copies have much lower fitness. Now, there are some drugs out that have just been developed that are using the CFTR protein, a normal one, and injecting that into individuals that have it, and they're extending their life. Okay? It's a disease that's common in Northern Europeans, just so you know. So we can actually look at this. Okay. So we can say how many CFTR alleles are needed for the person to be a carrier. Now remember, this is a recessive disease. So to be a carrier, how many copies do you need? Huh? 1. 1 to be a carrier. How many copies do you need for the person to have cystic fibrosis? 2.
2.
Okay. That's unlike Huntington's disease, where you only need 1 copy. Okay, so we'd have the same thing here. I'll just use N and M to signify normal. So you'd have N, N. Okay, here you'd have NM. Here you would have on the bottom and then finally here we'd have 2 M's. Okay. And remember, NM and MN are essentially the same genotype. It's just that they've inherited the different alleles from different parents in this case. So in one case, they've inherited the mutant allele from their father on the upper one, and on the bottom one, it was from their mother. Now, if a man with CF, cystic fibrosis, that means he needs 2 copies. So on the man up at the top, we have, he's CFTRM, CFTRM. Okay? And Mary's, what is the probability that their child, with an unaffected woman whose mother had cystic fibrosis? Okay? What's the probability that the child will have the disease? In other words, being MM. Okay, well, the probability of being a carrier, remember, inheriting only 1 copy, is 50%, right? So we can assume that if she's got 2 alleles, that one of them will probably be normal and one will probably be mutant. Okay? So we can say okay. Okay, so here we are. up at the top, we have a man who's mutant. The woman is normal. So we have N, M. And same thing across. The man, same thing. So okay. Now, the woman is a carrier, so she's a mutant. The man's a mutant. M. And again, so we have so what's the probability that the child will have cystic fibrosis? Huh? 50%, right? Okay. Now, Huntington's, again, is a very rare dominant disease. Remember, dominance has nothing to do with whether it's rare or common. It just has to do with how the phenotype is expressed. So if you inherit one copy from a dominant, that's a dominant gene, it will essentially cover up the recessive phenotype. So let's say we have a plant with red flowers that's dominant is red, and white flowers is recessive. If you have the allele for red from both parents, that means red, right? That's homozygous dominant. You'll have red flowers. You will also have red flowers if you have one red and one white, because the red is going to dominate the white. It's only when you actually have two alleles for white that you actually get a white flower. Same thing here. Now, obviously, that's a mutation. It may be good, it may be bad. It has nothing to do with whether it's good or bad in terms of dominance and whether it's common. You can have a recessive allele that's dominant, that is common because selection favors, let's say, the white flower at certain times of the year for pollination, okay? So remember, it has nothing to do, dominance has nothing to do with rarity or commonness. It only has to do with expression patterns. Okay, so Huntington's disease is a dominant mutation. It's very bad. It is not very common. It's a mutant allele that produces an abnormal enzyme, actually, a protein that doesn't break down another protein that clumps up on the nerve cells of the brain and causes damage. Onset is later in life, well, 40, 35, 40, something like that, 45. Okay. So say your father has just been diagnosed with Huntington's disease, a dominant condition. What are the chances that you will eventually develop the disease? Okay. Anybody, any guess? 50%. But let's take a look at why. So we'll use a Punnett square. Okay. We'll say the father is up on top. So he's got HM, and he's also got H normal. Okay? Because remember, oops, let's do that again. Okay, because you only need one copy for the disease. Probably if you had two copies, there would be earlier onset, like in childhood, but it would be very rare for that to actually happen. Okay, and then you've got a woman who obviously doesn't have the disease. In other words, she's normal. So okay. So the mutant is from the dad up here, and then normal. So here we would have an individual with Huntington's disease. Here, over here, we would have two normals. Here we would have on the bottom oh, okay, actually. Okay, and it doesn't matter. And then over here, we would have two normals again. Okay. So we would have, in this case, a 50% chance. 25% here and 25% there equals 50%. And then you would have two normal individuals that weren't, obviously, you can't be a carrier and not have the disease. So if you have the disease, you're a carrier and you have the disease for a dominant. So here we are. Okay. There'll be a number of problems like that on the next exam. There's a number of them. Let's see. Now, anybody ever hear of Gregor Mendel? Okay. Who was he? What? Huh? Pea plant? Yeah. What else about it?
Genetics, like the offspring generations.
Okay. What else was he? He was a monk. You see, you can be a monk and still be a scientist. This was, by this time, just so you know, the Catholic Church was no longer burning people at the stake for saying things that were contrary to the Bible. So, unlike Galileo, who was put in house arrest for the last 15 years of his life, the Catholic Church had finally come to realize that there is science, and if your science trumps belief. Okay, so he was a geneticist. In fact, he was the father of genetics. He was a Czech member of Czechoslovakia, now the Czech Republic. He lived in a monastery, and he was a biologist, and he started experimenting with pea plants. Now, pea plants, of course, you grow them for the peas. So he was more concerned with horticulture and agriculture and how traits were passed on. It turns out he actually had a copy of Darwin's On the Origin of Species, and he even annotated it, but he never connected what he was doing to Darwin and inheritance. Okay? And Darwin was unaware of him because he only published in basically a small agricultural journal that few people read. If Darwin had seen this, the big problem with evolution by natural selection would have been solved, because at that time most people believed in blending inheritance. That means it would be like paint. You have red paint and white paint, and you mix them together and you get pink paint. And so any trait that was beneficial would be kind of blended out, right? It wouldn't work. Whereas if it's particulate inheritance, and that's what Gregor Mendel actually discovered, that you have genetic and phenotype is connected because the genetic material is particulate. You either have this allele or you have that allele or some other allele, right? So he did a bunch of crosses in terms of flower color, in terms of what the pea itself looked like, whether it had smooth skin or crinkled skin, a whole bunch of different crosses that he did. And he worked out the genetics, and it turns out peas have a dominant recessive genetics. So it made it even easier for him. There are some cases where there's what's called equal dominance. In other words, if you put a pea plant with a red flower and a pea plant with a white flower, you'll get a pink flower. So it's not dominant or recessive, it's codominant. Okay? But anyway, in early 1860s, he did these experiments, and he actually figured out how genes and alleles were passed from one generation to the next. Unfortunately, his work, because it was in such a small journal, was lost for the next 50 years. And it wasn't until, or 40-something, it wasn't until the early 1900s that other people started experimenting with crossbreeding, and then someone actually found Mendel's paper in some obscure journal. When that happened, it solved Darwin's problem, because with blending inheritance, natural selection would not be strong enough. It would be kind of diluted out. But with particulate inheritance, it would work. And so there was something called the new synthesis. And it was a combination of Mendelian genetics and Darwinian evolution. And that's when kind of evolution came of age. Okay. Now, sometimes you will end up having, let's say, a trait, let's say in a pea plant. And the trait, let's say it's white flowers. White is recessive, red is dominant. Okay? So here we have, okay. On the left, we have an individual who has one copy, and this individual actually is recessive, so it's, let's say, white, white. So that's number 1. Actually. Okay, and then this is normal, okay? So we'll go back to oh God, okay. Okay, so if this individual pea plant has white flowers and it mates with a normal, you can get, let's see, hold on. Okay, I guess this is probably, I'm not sure, male or female, but anyway, this individual for the next generation, so there's generation 1, hold on. Generation 2 and generation 3. Okay. Now, in generation 2, from this side, we see we have a normal individual for 1, a normal individual for 2, and then a carrier for 3. Okay? I'm not sure how that happens. But anyway, now from this side, no one is a carrier. So there must just have been, hold on a second. Oh, that's what it is. Okay. We don't know what 3, 1 and 3 is, but they must be heterozygous. Okay, so okay. So when you have a recessive trait in the first generation that's expressed, in the second generation it's lost, because remember, for recessive you need 2 copies, right? You only get 1 copy, it's a carrier, but it doesn't show up phenotypically. In other words, it doesn't show up as a white flower. Any individual is going to have a normal red flower, dominant, and white flower, recessive. So they'll be carriers, but they won't actually express red flowers. And it's only shows up later in the third generation when you have 2 individuals who are carriers who mate, and then you end up with 2 individuals who are red flowered. Okay? So they've inherited a copy from mom and a copy from dad. Or and their parents were each heterozygous. They were red and white. Okay? So recessive autosomal traits appear equally in both sexes. That means the autosomes has nothing to do with the sex chromosomes. But with recessive autosomal traits, you can have a situation where one generation expresses the trait. In this case, it would be white flowers. The next generation does not, only expresses red because there's a mixture of pure red and also heterozygotes red-white. And then the third generation, because of those heterozygotes, you can end up with white again. So in a dominant situation, you will never have a situation where traits skip a generation. It will only be recessive. Okay. So it's a failure of chromosomes to separate during cell division. So it results in gametes, they're haploid, that have one too many or one too few chromosomes, and that most embryos that result from such gametes will die before birth. In other words, it's a lethal mutation. Natural selection wipes it out, right? It's got a lethal mutation, no offspring. Okay. This is a cell. And we've got chromosomes that line up along the metaphase plate. Normally what happens is, as meiosis goes along, you end up with two daughter cells, or two gametes rather. Let's see. No, actually it's in meiosis 1, so two daughter cells initially. And each side has hold on a second. Okay, has chromosomes on each side. Okay. But sometimes you end up with 3 chromosomes on one side and 1 chromosome on the other. That's called non-disjunction. Okay. However, there are some individuals where there's non-disjunction and it's not lethal. But they usually have some problems in terms of phenotype. So trisomy 21 right here, it produces an individual with 3 chromosomes for chromosome number 22. Or no, chromosome number 21. Sorry. Instead of 2. So they've got 3 copies. And then you end up with an individual with Down syndrome. Some Down syndrome individuals, well, they have a phenotype where they look a certain way in terms of facial features and other things. Some of them are pretty high functional, but they still have mild, let's say, retardation. Others are much more seriously developmentally disabled. Okay? So, now if you had an individual that only had 1 chromosome instead of the 3, you would definitely have a lethal, where the embryo would die while it's developing, early in the pregnancy. Okay, so now we're on to sex chromosomes, and sex chromosomes they act a little bit differently. first of all, for humans and for mammals in general, females are XX. So you inherit 2 copies just like before, and if you inherit 1X from your mother and 1X from your father, you end up with a daughter. If on the other hand, you have an X from the mother, remember females are always XX, and the man donates a Y. Okay, so it's XY, that means that you have a boy, a male baby. Okay. So the males in this case are what's called the hetero, means different, gametic sex. Okay, so okay, and the females are homogametic. It turns out in birds it's different. They don't have XX, they have ZW, but it's the females that are the heterogametic sex. So a male is ZZ, and a female is ZW in birds. So it doesn't have to be always that it's the man who's heterogametic. Now, has some consequences in terms of which side is the heterogametic sex. If there's a mutation in an autosome, let's say this one right here, actually. Okay? And it's a recessive mutation. What will happen is that individual has a 50% chance of passing it on to their offspring. And then the other individual doesn't have that, just normal. So often what happens is there's something called a rescue effect. Haplosufficiency. So even if you only have 1 copy producing the protein, it's enough that you have normal function. So like with the pigment red in a plant, in the flower, even though if it's a dominant allele, all you need is 1 copy for red. So even if you have red-white, you still have a red flower, okay? Now, sometimes in autosomes or somatic cells, you get a situation where there's haploinsufficiency, and you actually need, for normal function, 2 copies of the right allele. So in this case, let's say white is recessive, you would end up with haploinsufficiency of pink flowers. Okay, there's pigment from the red gene and pigment from the white gene mixed together. They're particulate, they're not blending, but that's what you end up. Now, getting back to sex chromosomes. When you have rescue effect in a somatic cell, you end up with normal function, okay, if it's dominant. But remember, if you have, in sex chromosomes, let's say you have 1 normal X and 1 mutant X, and the mutant X gets donated to the child. If the male donates an X and you end up with a daughter, they're just carriers. It's no biggie. They'll still have proper function. because it's dominant trait. But if the male donates the Y instead of the X, there's no rescue effect. So, being the heterogametic sex, you will often find genetic mutations on the sex chromosomes will predominantly affect the male and not the female. Okay? In fact, there's a whole slew of, I guess that's it. We'll get back to this, but there's a whole slew of genetic diseases that are sex-related, like blue-green vision, being colorblind, other things. So that's where we'll end.